Whose win is? No one has.
Okay. All right, folks. Time to restart. Okay. Hello, folks. Hey. Okay. All right. Well, we're going to start now looking at nucleic acids. Now, we're not going to mention much about DNA. But first, I want to make a little summary chart of the differences between DNA and RNA. That's a quick and dirty thing. And then we'll look at the functions in a little bit. Both of these are nucleic acids made, and most of them exist, especially if DNA exists in the nucleus of cells. DNA uses the sugar deoxyribose, which is like ribose, except instead at one particular point in the molecule, instead of having a hydroxyl or OH group, it's got a hydrogen there instead. It's missing one oxygen relative to ribose. RNA, ribonucleic acid, uses ribose. Bases of DNA are adenine, cytosine, guanine, and thymine. Thymine is unique to DNA. With RNA, it's adenine, cytosine, guanine, and the very similar uracil. So, for instance, if you want to detect newly synthesized DNA and not worry about newly synthesized RNA, you add some kind of labeled radioactive or fluorescently labeled thymidine, the nucleotide that goes into DNA. And it will get incorporated only into DNA and not RNA. Likewise, you could use labeled uridine and you find the RNA production, not the DNA production. Okay, now, so those are the bases. And uracil and thymine are very, very similar. Okay, now, the other thing, DNA, the vast majority of DNA is a double-stranded molecule. Our classic double helix, you've got our sugar phosphate backbone structure here. The bases stick off of that structure, and you get the hydrogen bonds between, say, adenine and thymine, cytosine, guanine, adenine, thymine, so on. Okay, so it's a double structure. Now, hydrogen bonds are relatively weak, so to break the two strands of DNA in half, separate them out, you merely need to heat them up. Generally, close to the boiling point of water is more sufficient. If you have DNA with lots of cytosine, guanine, or CG base pairs in there, since they make 3 hydrogen bonds, those are a little more resistant to breaking and have to heat them up. Pretty warm, pretty close to the boiling point. If you have lots of AT-rich regions, those separate out easier. Enzymes, of course, in living cells do the separation. We don't heat things up. But that popular technique for copying DNA from little small amounts like a dress stain on a White House intern shirt, remember that? Remember that one? Okay, well, those little tiny amounts of DNA copied by a technique called PCR, the polymerase chain reaction. What you do is repeated heat-cool cycles to separate the strands of DNA, and then as they cool down, the DNA polymerase enzymes copy each strand, and then things cool down, and you start the cycle over. So the DNA content doubles every time you run a cycle, and you can run them 30, even 40 times with modern technology. So you're talking about a billion to a trillion-fold amplification. So that's why people have gone to jail on the basis of a little tiny bit of saliva, dried saliva on a cigarette butt at a crime scene. That's why presidents have gotten impeached from a 2-year-old stain on an intern's dress and stuff like that. Okay, so DNA is normally a double-stranded molecule. Now RNA is generally a single-stranded molecule, but there are regions in RNA where you may have opposite or complementary bases that can form a double-stranded region. For instance, let's suppose we have an RNA molecule. Here we have A over here, and twists around, loops around, and over here we got a strand that has U. Oops, going over here. Okay, we can get hydrogen bonds forming here, so we'll have this double-stranded region, and then it diverges again. And that's a common kind of thing. We often call these in RNA molecules, these what we call the stems. the hydrogen bonded regions, double-stranded regions, and areas that are not, we often call the loops. Each different kind of RNA, especially large RNAs, have complex and reproducible stem and loop structures. If you look in the book, for instance, you get to see that structure of the large RNA that's found in ribosomes, the biggest of the ribosome RNAs. And you see a very complex structure of multiple stems and loop regions. Okay? Now, it turns out those regions are actually functionally important because many times the stems are used as binding sites for various kinds of proteins. So these are not just pretty pictures, they actually have functional importance. So many RNAs do have double-stranded regions. So those are major differences between DNA and RNA, at least in the chemical sense. Okay, what about the functions of these molecules? We'll take a brief look at DNA first. However, this is not a genetics class. You've already had a genetics class, or many of you have, and some of you may take our molecular biology class. Okay, in short, DNA, of course, is the storehouse of genetic information and everything on this planet that we can call life. Now, viruses, most scientists would not classify them as truly alive because they're incapable of synthesizing their own materials and replicating themselves on their own. What they have to do is get into a host cell and basically hijack the host cell's machinery. So most people do not consider viruses alive because they cannot replicate independently. They have to be in a host cell because only the host cells have the machinery in order to do the replication. They'll produce proteins and code for proteins that will assist in that process, but they can't do it on their own. Okay, and many viruses, such as the pox viruses and the herpes viruses and stuff, those have DNA genomes. Okay, many other viruses like the flu virus, most rhinoviruses, those ones that call colds, polio viruses, those have their genetic information in the form of an RNA molecule. But in terms of living organisms, from those primitive archaeans and bacteria all the way up to us, DNA is the storehouse of genetic information, or what we call the genome. The whole genetic blueprint, the instructions for making a cell, or for that matter, an entire organism. Think of this, every single one of our cells has packed into that nucleus that's just a couple micrometers across in most cases, has the information needed to specify literally a new you. And of course, that's the process of cloning. Now, nobody's been able to clone a primate so far, although people have cloned dogs, cats, cattle, rats, mice, and some frogs and some other kinds of organisms. I think they clone chickens and stuff as well. They've been able to do that. Nobody's been successful on cloning a primate yet, which is probably a good thing because then you're going to get, Kim Jong-un or whatever in North Korea want to make more copies of him. So I think one is more than enough. We really don't need multiple copies of him in this world. But at any rate, the fact that you can do cloning where you take the nucleus from a somatic or body cell and stick it into the properly prepared nucleus of an egg cell, and it will make a genetically, if you're really, really lucky, fails most of the time, and make a genetically identical copy of the whole organism. That tells you something. Every single one of our cells has the information for a new use specified in the nucleus. And that's in the form of DNA. Now, human beings and most vertebrates in general, how much information do you have in there? Well, for most vertebrates, it's about 3.2 billion pairs of bases. And that's enough to code for somewhere between 20 and 25,000 separate genes. But as we're going to see, there are many other types, sections of DNA that have other kinds of instructions that are not directly involved in protein coding genes. So that's our genetic blueprint. Now, why does life on this planet use DNA instead of RNA? It's been hypothesized that early proto-life, well before we see anything in the fossil record, may well have used an RNA-based genome as some viruses do today. The problem, though, with RNA is nowhere near as stable as DNA. If you work with nucleic acids in the lab, DNA is pretty tough stuff. You can beat on it pretty good. RNA, you look at it funny and it falls apart on you. It's an unstable molecule. DNA is much more stable and much less prone to be broken down than RNA, which is why even if there was a, quote, RNA world 4 billion or so years ago, life nowadays universally uses a DNA genome. Now, one or two other things about DNA. How is that specified? How do we have that information? Now, just writing out, if you were to use the same text and font size in your textbook and write out 3.2 billion characters, that much information would fill about 600 copies of your textbook. That's how much information is packed in every one of our cells. And I'm not even sure if you could fit that on a CD-ROM. You might have to go to a DVD or even a Blu-ray to pack the information of a human genome into that. Okay, so it's an awful lot of information packed into a very, very small space. Now, how is that information stored? It's basically very much like a language. The sequence of bases on one strand of DNA are going to represent something. So here's 2 strands of DNA. I'm not going to let's take them apart, but here we have. Okay. Oops. Get a little closer here. Okay, here's 2 strands of DNA. Now, what happens is you look at one of the strands and read the sequence of bases. And that sequence of bases tells us something. In a sense, it's like a language. Let's suppose if I write something in the English language. Okay, if anybody doesn't know, we will have a test tomorrow. Okay, now what is that? It's a bunch of squiggly chalk lines on a blackboard, but it has some meaning here. What we do here, we have a number of different letters, 26 letters in the English alphabet. Those letters, certain combinations of letters form words, and those words represent certain concepts, like test. Everybody knows what that means. Tomorrow is referring to whatever day is immediately after today, and so on. We also have punctuation marks, like a capital letter at the beginning of the sentence, a period at the end of the sentence, and so on. Okay, now, the language of DNA is very similar, except we only have 4 letters, the 4 different types of bases. And, at least in the parts of DNA, what we call a gene that actually carries the instruction for protein, each sequence of 3 bases, what we sometimes call a codon, represents a particular amino acid in a protein. And then we have other stretches of DNA that act as punctuation marks, things that we could say the transcription start site, something we call the promoter. Those provide the cell with the information on when that gene is supposed to be used and activated and how much it's supposed to be activated. So we have these kind of things. And like a language, now imagine if I read this sentence the opposite direction. It would come out as gibberish. I'm not even going to try that. So we read English in a particular direction. If you happen to be Dr. Hamasu or Dr. El-Hamdani, they're both fluent in Arabic because they're Muslim, and Muslims are supposed to know Arabic and supposed to read the Quran in the original language, so you don't have to worry about translation errors and things like that. Okay, now Arabic is read the opposite direction. We read things left to right. Arabic is read like right to left. So here again, you read a particular sequence of DNA in one direction, but that isn't always the case. You might have another gene that is read this direction. And sometimes this strand of DNA is going to be what we call the coding strand, and other times for other genes it's this strand of DNA. And sometimes you'll even have overlapping sequences where part of a gene is over here and stretches to here, and part of another gene ends here. That occasionally happens too. It's not too common in higher eukaryotes, but is known. So in some respects, the language of nucleic acids, the information, is very much like the information stored in some kind of alphabet, some kind of language. Okay. Now, another interesting thing about DNA, clearly, if you packing the information in 600 copies of your textbook and packing that into something that's just a few micrometers across, the nucleus found in all of our cells, that's a tremendous amount of information storage. Take the sequence of the human genome, you probably would have to put it on a DVD rather than a CD. And yet they're putting that much information in something that's just a couple micrometers across. There's actually, because the tremendous capability of packing huge amounts of information in small spaces that the DNA molecule exhibits, there are even currently a few high technology companies, and this is just fledgling the technology, but investigating the possibility, could DNA sequences, artificially made DNA sequences, be used as an information storage media for our own technology? Now they're just taking little baby steps on this, but 50 years from now or something, that could be a totally different story. If you could do that, picture this, having one little plastic DNA disc that not just has a film on it, has every film ever made in the past 10 years packed onto that single disc. That's the kind of information storage capacity that DNA has. So there is some work on that. There'll be a long time before it actually ends up in your local Walmart, but the potential is there. Okay, so that's what we see with DNA, the storehouse of genetic information. And in eukaryotic cells, we keep the DNA safely inside the nucleus. Prokaryotic cells, which don't have the nucleus, the DNA is generally in the center part of the cell. It's not in a specific membrane-bound compartment, but usually the DNA of bacteria and archaeans are packed towards the center. Okay, well that's DNA. Enough on DNA.
Probably good.